And welcome to the Wednesday afternoon lecture series. I'm Michelle Antoine, the acting chief of NIAAA's section on neural circuits, and it's my honor to introduce today's speaker, Dr. Guoping Feng. Dr. Feng's research has provided remarkable insights into how synaptic abnormalities contribute to the behavioral manifestations of neurodevelopmental disorders such as autism spectrum disorder, Rett syndrome, Williams syndrome, obsessive compulsive disorder, as well as psychiatric conditions like schizophrenia. For those watching remotely, we encourage you to participate in the Q&A session of today's presentation. You can do so by clicking on the button just below your videocast window that says Send Live Feedback. Click on that, type in your name and question, and we will relay this to Dr. Feng at the end of his lecture. You can submit a question at any time, and I hope you do so. So for those of you in Lipset Amphitheater today, you can use the mics on the left and the right of the room to ask questions. Also, we are offering continuing medical education credits for this presentation. So the CME code for today is 57683. I'll repeat that, it's 57683. So our speaker today is Dr. Guoping Feng. He is the Associate Director of the McGovern Institute at the Massachusetts Institute of Technology. He's also the James W. and Patricia T. Poitras Professor in Brain and Cognitive Sciences. He's the director of the Hock E. and Tan and K. Lisa Yang Center for Autism Research. He's the director of Model Systems and Neurobiology at the Stanley Center for Psychiatric Research at the Broad Institute. Dr. Feng received his PhD from SUNY at Buffalo under the thesis mentorship of Linda Hall, where he worked on G-protein coupled receptors, I believe GPCR60 from Drosophila melanogaster. He then did a postdoc with Josh Rosines at Washington University School of Medicine, where he continued to hone and deepen his knowledge on synaptic function, but in vertebrates. In 2001, he moved to Duke University, where he became an assistant professor in the Department of Neurobiology. And while there, he established a powerful blueprint for how we as neuroscientists can use genetic rodent models to replicate some complex human diseases, the symptoms of complex human diseases like obsessive compulsive disorder. And he demonstrated that treatments that are effective in humans could be similarly applied to reverse these symptoms modeled in mice. Although his work illustrated how to establish the critical steps in modeling disease, he established our ability to produce phase validity, construct validity, and predictive validity in mice. His framework laid the foundation, of course, for the next essential step, which is connecting the specific abnormal behavioral symptoms to the underlying genes and synaptic mechanisms. And that groundbreaking work was published in Nature in 2007. So as a young graduate student at the time, I was deeply inspired by Dr. Feng's work, and I continue to do so today. And in fact, many of us in this room continue to apply Dr. Feng's step-by-step, streamlined approach to tackle some of the most difficult and complex biological questions in our own work towards understanding disease mechanisms and identifying new treatments. In 2010, Dr. Feng joined the McGovern Institute, where he remains today. And since then, he has further refined his already rigorous framework to explore the role of mutations in the SHANK3 gene, and once again linking gene function and synaptic alterations to autism-like behaviors in mouse models. His research has also helped affirm the promise of gene therapy as a treatment strategy for neurodevelopmental disorders. Dr. Feng has continued to push the boundaries of neuroscience by extending his research framework to non-human primate models, an exciting and ambitious step that you'll hear more about in his talk. His work continues to shape how we understand and treat complex human diseases. Dr. Feng has received numerous high-profile awards along the way, including the McKnight Technological Innovations in Neuroscience Award, the Hartwell Individual Biomedical Research Award. He was elected to the National Academy of Medicine in 2003, the American Academy of Arts and Sciences in 2019, and the National Academy of Sciences in 2023. The title of Dr. Feng's talk today is Dissecting Neurobiological Mechanisms of ASD from Genes to Circuit. Please help me welcome Dr. Guoping Feng.
Thank you, Michelle, for a very generous introduction. It's a real honor to be here and to be in this powerhouse of research. And since I was a graduate student here in U.S., I always admired the unbelievable work done here. And actually, at the end of my talk, you will see I actually still follow some of the early discoveries and from NIH campus. Of course, I'm always very grateful for the NIH supporting all the extramural programs that we rely on this. I think we are more and more need this at this time to push our research into benefit of patients. And today I'm going to talk about some of the research. I'm thinking how can we build on our basic research in the past many decades and eventually can potentially help patients. This is all one of the main goals of NIH research and many of our scientists' goals and also our neuroscientists' goal. So this is my disclosure already showed by Michelle's slides. And so in the past 20 years, one of the biggest advances in brain science, brain disorder research is the identification of risk genes. Because as a scientist, for brain research, unlike cancer, which is also a very difficult problem, it's very hard for us to get live brain tissues, the whole brain, to study. So we heavily rely on animal models, right? And so to be able to use animal models, we need, generated models, and genetic information is one of the key things that we can actually engineering, and we can put the human mutation into genetic models. Use autism risk gene as an example. We actually have been. Let me see. I can no. And so in the past, 2 decades, we have identified many groups, not in my lab, but many other groups, and identified hundreds of risk genes. Some of them are monogenic causes of ASD genes. And with this genetic information, we can generate with the new technology, especially genome editing technology, we can generate many kind of genetic models, including cell models, rodent models, and primate models, and then use these new technologies like CRISPR and circuit manipulation, and especially single cell transcriptomic analysis. We can help us understand the neurobiological mechanisms. The hope is with this kind of study, we can eventually explore therapeutic strategies. And so for use ASD example, many of the genes actually are ASD risk genes affect the synapse, play an important role in synapse development, function, and plasticity. This is a slide I borrowed from Thomas Bourgeois' review, and you can see from the from these excited synapse, inhibited synapse, these brown colors are the proteins important for synaptic function and development, but also risk genes for autism. And So my lab, like many labs, are very interested in the development, function, and the dysfunction of excitatory synapse, especially in the postsynaptic site. So many labs in the previous 3 decades have contributed our understanding of a molecular architecture of excitatory synapse, postsynaptic density, including Mary Kennedy, Morgan Shen, Hugenel's lab, and Boger's lab in Germany, and David Bress, and many other labs. I just list a few of them. So in this very small structure, postsynaptic density, which is, the width is about 2 microns and the thickness is about 200 nanometers, they are actually identified about 1,500 type of proteins. And they have, density impact. That's why I call it postsynaptic density. You can see in the EM without any staining. And how these such a macromolecular structure is assembled and dynamically regulated is always a very interesting phenomenon to many of us, especially to us who study synapses. So one of the mechanisms for this assembly is called scaffolding protein. So one of the scaffold is PS95, SAPAP, and Shank. They build a scaffold. Each of these proteins have multiple protein interacting domains. And from one end, PS95 proteins interact with membrane proteins on the postsynaptic side, including AMPA and MD receptors and ion channels and adhesion molecules. On the other hand, you can bring many signaling molecules to the synapse, to postsynaptic site very dynamically. And on the other end, with Shank, not only bring a synaptic molecule to it, a signaling molecule to it, but also it can interact with cytoskeleton interacting proteins. So it play a role in not only the function, but also plasticity, both in the signaling and functional plasticity, but also structural plasticity. And so one of the proteins we are really interested in, and many others are also interested, is the Shank family of proteins. Shank is considered a master scaffold for the assembly of the postsynaptic site at glutamatergic synapses. But more importantly, one of the Shank, called Shank3, is a mutation of Shank3 is a monogenic cause of very severe autism with intellectual disability. And this is also the main gene in the deletion of in the Phelan-McDermott syndrome, which is originally identified by Dr. Phelan and Dr. McDermott, and on 22q13.3. And these patients are very severe. They have hypertonia, development delays. They have severe intellectual disability. Most, if not all, patients can live independently and cannot live independently. And they also have severe impaired language development and, of course, autism spectrum features. And they also have many other comorbidity issues, including sensory issues and seizures and sleep disorders. So our original idea was this is, because it's a monogenic, we can make models and then try to understand what is the molecular change that we can identify, then we can develop therapeutics drugs to treat this condition because it's so severe. So one of the features of this gene is it's highly expressed in the striatum. There are 3 Shanks, and only Shank3 is highly expressed in the striatum. And so our assumption is it will affect the corticostriatal synapses and affect the corticostriatal thalamocortical circuits, which dysfunction has been strongly implicated in autism and other neurodevelopmental and psychiatric disorders. So we initially, I'm going to go through very quickly, we initially made a knockout of Shank3. I found indeed it's a scaffolding protein. You knock out the Shank3, you will lose many other synaptic proteins in the postsynaptic side, including as a scaffolding protein and NMDA and AMPA receptors. It also affects the function, reduce the synaptic transmission, mostly synaptic minute frequency, but also amplitude. They also affect the number of synapses in the minimum spine neurons. We focus on the analysis in the corticostriatal synapses and the spine numbers and the medium spine neurons in the striatum. So these animals also have many behavior, in the homozygous knockout. In patients, they are all heterozygous, but in mice, heterozygous have very little phenotype. So we use homozygous to study the function of Shank3. It also has many abnormal behavior. One is repetitive compulsive grooming. They grooming hair off and skin off. And they also have in 3-chamber assays. Jackie Crowley originally, developed here at NIMH, and also have a social interaction deficit. They are also sensory oversensitivity, the anxiety, reduced locomotion, impaired motor learning. So many of these phenotypes, we cannot say identical, but it's relevant to autism spectrum disorders and their comorbidity conditions. So can we identify converging mechanism that can allow us to develop a drug? So we start to look at the molecular and cellular defect in the animals. I'm not going to go through all the studies. I've just summarized you one. What we found is for each phenotype, there is a different cell type involved and different defect involved. So, for example, libellary compulsive behavior, we found it's reduced the neuronal activity of indirect pathway medium spine neuron, which is D2 positive medium spine neuron in the striatum. If you correct the defect in D2 medium spine neuron, you can actually correct the repetitive grooming behavior. And for the social interaction, many labs studied. My lab also studied with a former postdoc in my lab, and found that it's mainly their study, found that the reduced activity of excitatory neurons in the ACC is the cause, at least one of the cause of the social interaction deficit. If you increase the activity or knock out SHANK3 only in these neurons, you can provide direct evidence that this group of neurons at least is involved in this social interaction deficit. We also study sensory oversensitivity in collaboration with Chris Moore's lab at Brown University, showed that reduced activity inhibiting interneurons in the somatosensory cortex is responsible for this sensory oversensitivity and sensory overload kind of phenotype. So put them all together, this is a single monogenic cause, right? So it's even for a monogenic cause, you have different circuits, different cell types in different brain regions for different phenotypes. And imagine this is polygenic. We even don't know how to model them. So it seems like it's really difficult to identify a converging mechanism from multiple models of autism that find a therapeutic target. So it's not surprising because for SHANK3 and many others, including Fragile X, it's a translational regulator, affect 800 genes by Bob Darnell's lab at Rockefeller University. And Fragile X, Heritology and many other labs show that at least 300 genes are affected by a transcription. So for SHANK3, it's a scaffolding protein. It interacts with many different proteins. And so it's not surprising that, can you imagine, in different cell types, during different development time, it will require different signaling mechanisms. So it will be very difficult to find a converging molecular mechanism in our view. So all these studies, my lab studied, many other labs studies show that there are many pathways that have been disrupted. So then the question is, if it's so complex, how can you find a therapeutic target that can benefit many of the autistic kids? And so even for a monogenic cause, how can you treat all the different symptoms that are caused by different mechanisms? So that made us to think about to change the strategy. And we think that many of us, including myself, think that maybe we use different strategies. For the monogenic cause, maybe gene therapy with the new technology development might be the best way, or at least one of the best ways to directly correct the genetic defect that could be potentially a cure for these monogenic disorders. On the other hand, most of autism or most of psychiatric disorders are not monogenic. They're caused by polygenic factors plus other factors, including environmental, many other factors, and developmental insult. All these could contribute to it. For these, we think that one of the approaches could be by understanding neurocircuits that cause a particular most debilitating symptom, and by correcting that circuit defect, we might be able to dramatically improve the brain function, behavior, and patient's life. So I'm going to use two examples, use SHANK3 as an example to illustrate how we might be able to make a progress in these two fronts. And neurodevelopmental disorder, especially autism, these are neurodevelopmental disorders that their defect may be developmental, may be irreversible. So some of the early work, landmark work by Andrew Bird showed in Rett syndrome, Actually, if you provide a gene later on, it can reverse everything. So, but I think each gene is different, depends on where they are expressed and when they are expressed, right? So I think for each gene we have to test them. So we tested for SHANK3. We made a mouse model that use double flux, basically reverse several exons, encoding the PDZ domain, which is one of the key functional domains in the SHANK3 protein. And so these mice actually grow up like a knockout. They have basically all the phenotypes I described before. Then because of the flux is Cre-dependent, you can use a tamoxifen-inducible Cre cross, then feed the animal with tamoxifen at different time point postnatally, and to see at which time point you provide a full SHANK3 gene can rescue this phenotype. So what we found is that in adult, if you turn the SHANK3 gene back on in adult, you can actually correct some of the defect. For example, repetitive grooming. This is before the tamoxifen. This is before the tamoxifen feeding. You see the grooming, their skin off. After the tamoxifen feeding, the lesion healed, the hair grow back. The only difference is the hair grow back white. This we think is related to the tamoxifen. Tamoxifen is a breast cancer drug. Actually, some of the patients you can see online, you can check. Some patients, why my hair become white? So you can see that this can be reversed. We also found that surprisingly social interaction also can be reversed. However, many things we cannot reverse. Anxiety-like behavior, sensory oversensitivity, motor defect, none of them we can reverse in adult. However, if you turn on the SHANK3 gene much earlier, postnatal, 3 weeks postnatal, P21, or 2 weeks, you can reverse almost everything. So that gives, there might be this is consistent with all the great work done by visual system critical period. Once you pass the critical period, whether it's a visual, language, other things are very hard to modify. So, but at least this tells there might be a postnatal window for gene therapy. If you can turn on the gene, within a certain period of time postnatally, you might be able to correct most of the things. So, of course, for human patient, we cannot use this genetic approach, but we can try gene therapy. So we decide to use gene replacement gene therapy. The reason is most of patients, at least many of patients, are large deletions. Right now, genome editing technology, which have great promises for medicine, cannot do that easily. And also, even the point of mutation, they're all over the gene. Like each mutation, there's not a single mutation account for many patients or most patients. So we decided we want a system that can treat for all, vast majority of the patient. And so we decided to use gene replacement and postnatally. But right now, the main way to deliver into genes into the brain is use AAV. But AAV has a packaging limit. So this SHANK3 is way too big for AAV. So we decided to make a mini SHANK3, which actually contains some of the most important domains and still functional. So we tested in vitro, we tested in vivo, we use electrophysiology and synaptic localization to show it is still functional. And use biochemistry to show it goes to PSD. So with this approach, we actually use the AAV capsid that can pass BBB, so you can do IV injection, developed by Ben Deverman and Viviana Granado at Caltech. Ben now is at the Broad Institute working with colleagues now. So we can use this, then we can, because of gene therapy, we can inject at different time point. Postnatal 0, P7, P14, P28, and you wait for 2 months, then you can do biochemical tests, you can do electrophysiological tests, and you can do behavior tests to see at which time point, first, can you rescue them? Second, at which time point you can rescue them? What we found is in all these time points, you can almost rescue almost all the phenotypes very clearly. So this is an example. So corticostriatal synaptic function. This is a field recording. Black is the Y type. Red is the SHANK3 injected with AAV GFP. And then the blue is the Shang-3 injected with AAV mini Shang-3. You can see that restored cortical striatal function. This is one of the behavior, social behavior. The knockout there's no preference for social compared to the empty chamber or object, but the gene therapy rescued the behavior. So that gives us some hope that maybe this gene therapy will work. The issue for us at that time is to move this to humans is a real challenge, because gene therapy, unlike drug testing, right? If you take a drug, it has a side effect, a severe side effect, or not effective, you can stop it. With the AAV, you inject it, you can never take it out. So we thought we probably need a better model, or at least another model closer to human to test them, right? So we all know mice are great models for biomedical research, is evolutional for the past many decades. But there are also significant differences, right? Especially now with there are anatomical studies, connectivity studies, and single cell transcriptome studies and cell type studies. There are still many differences. So it will be really good for us, especially to study social cognition or other high function. We have a non-human primate model. So that time with many support for our donors, and now more recently with the Brain Initiative and NIH support, we are really grateful for the Brain Initiative especially. And we generated a marmoset model. Marmoset is a small non-human primate, a New World monkey. They are not as close not as close as the macaques to the humans, but they also have a significant advantage. They are very small. Their reproduction is much faster. They sexually mature in about a year and a half, and they give birth twice a year. Each birth gives twin or triplet. So that if you make transgenic animal, you want to be really sexually mature faster. You really want to breed faster, so you don't have to wait for 20 years, have a colony, you can study them, right? So our first model was actually Shang-3. We made a small deletion, mimic the human condition in exon 21, and we'll lead a frame shift. And we also made a single G insertion, which is also commonly found in several actually patients now. This is actually one of the most common insertions in Shang-3 mutation in patient. And so now we have a significant number of F1s, the pure heterozygous. So in marmoset, we don't study homozygous. We mimic human conditions, the human type of mutation, and use heterozygous to study them. So then the other advantage is marmoset is very small. Their cage is very big compared to their body size, and they live in a family. So this is, you can see that we develop a machine learning-based behavior tracking, and we can track them really well. Now you can just put a camera on. You can track them for days and weeks or months, and then because it's an automated behavior quantification, you don't have to spend a lot of time. Computer will do all the work for you, right? So I want to just give a couple examples how we study them and what does it look like, right? So with this kind of program, you can track their behavior. So for example, there are two marmosets in their home cage. All these studies are in home cage, and you can look at their fraction of time they stay together, which is social. They sometimes groom each other. They will just stay and looking together, like coordinated looking, and are just sitting there to be friends. And there are also fraction times they are apart from each other. They're running around, they doing their own things. And anyway, Y type. If two are Y type, whether it's a young animal or adult animal, they always have two peaks, right? Near each other, apart from each other. But if one of them is Shang-3, what you will see is this near each other peak is gone. It's almost like you go to a bar. Right? You want to meet someone. You got a drink. You sit down with someone. You start to talking or social. If that person never responds to you, you're not going to stay there very long. Right? You're going to find somewhere else. So it's a really interesting phenomenon you can see from this automated behavior tracking. And there are many other things you can do. For example, moms love iPad. I don't know whether they love iPad, but they like touchpad. So you put a touchpad, they all go in front, right? And you teach them very quickly touch a particular spot, you get a reward. They love marshmallow. So we give them a really teeny tiny marshmallow. So they will always go touch it and go back, get a reward, then go touch it again. So you can see, when you have a touchpad, if a touchpad is a blue square, they all stay in front, right? Whether it's a Y type or Shang-3. If you change this blue square to a marmoset imaging, Wild type doesn't care. Want the reward. Mutant, they actually stay back more. And they still will go get rewarded, but they don't want to stay there all the time. They just go there. So it's a really interesting phenomenon. Even a phase of marmoset, they don't like to stay there all the time. And you can also test it with animal, right? So this is a small cage. Put it inside the cage. Of course, they cannot come out. But if they're all wild type, you can see wild type all want to come to look. Who is the new kid on this block? Like, just like a human behavior. And you want to, investigate. But if it's a mutant marmoset, they are actually really scared of stranger marmosets. They will just scream and climb on the top of the, of the cage. That's usually their safe place, like trees, all this. So you can see there is a significant response to new things, including new marmoset. Right? So with this kind of assay, we can do a lot of things. Naturalistic behaviors, because they're always in their home cage. That you can do iPad to do cognitive testing, motivational, social, a lot of things. And you can also do induced behavior. I think because marmosets are very heterogeneous. They are not inbred. They are completely outbred. We do whole genome sequencing for all the breeding marmosets, make sure they are not inbred. So their behaviors are very variable, right? So just like humans. But we found that if you do compare before and after stimuli, that's the most robust phenotype you can find. So this is actually one of the most important assays we do. And of course, you can do basic function, whether it's feeding, their gait, or their sleeping time, all these things you can do, right? So with this kind of assay, now we can test, will your gene therapy improve any of these issues, right? So we did 2 groups of gene therapy. One is, we have a large wild-type control. We track them for long time, so we know the baseline. Then we have a group of Shank's-3 without gene therapy. And one group of Shank's-3 started with 9 months gene therapy. 9 months is about 10 to 12 years old kids in development. And then we have another group started with 3 months old baby. 3 months old baby, about 3-4 years old human baby. So then we wait for 3 months. This is the IV injection. We found that with a separate marmoset, with IV injection, with this particular capsid developed in collaboration mostly by Ben Deverman's lab at Broad right now, and it can. This is specific to marmoset. Unfortunately, it doesn't go to even macaques, so I don't think it will work with humans. But it works really well with marmoset. And you can in fact use synapsin promoter. Is in fact about 15 to 20% neurons in the brain with IV injection. And so we wait for 3 months, then every 3 months we test their behavior. Right? So, and what we found is, we found how do you, the most robust, because you cannot use 20, like mice, 20 monkeys for wild type, 20 monkeys for control, 20 monkeys for gene therapy. So we only have 8 monkeys, right, for the gene therapy. 4 from 3 months, 4 from 9 months. So we found the most robust way is to compare their behavior to their cage mate, wild-type cage mate. Because monkeys living in different rooms, different cages, if one cage mate is very hyperactive, the other will affect the others. They were more active. One is more anxious, and then if someone come to change the cage, make a noise, the whole room will be affected, but the other room will not be affected. So we found its most robust way is to see the monkey's behavior, how correlated with the wild-type control in the same cage. So, for example, these 2 are in the same cage. If you look at their behavior, you subtract them, very little difference. These are 2 wild type. But in the same cage, if one is a wild type and one is Shank's-3, you can see they're very different. So now you can see, can you reverse this into become like this, right? So we did a lot of assays, tracked many hours. This is 5,000 sessions. It's almost like, ADOS review, ADOS question, 200 questions or whatever number of questions, and you click all the things. What we found is that this is wild type, right? Their correlation with the wild type cage mate. This is Shank's-3, way less correlated behavior with the wild type, and gene therapy. Overall, you can see they corrected to the wild type level. Then you can go in, look at each individual behavior, which is corrected, which is not corrected. So from behavior point of view, you can see they are, for wild type, they are widespread. For monkeys, widespread. But you can see on average, Shank's-3 animal, this is touch screen, right? And they are less correlated with compared to wild type. Gene therapy can restore to wild type level. This is the marmoset face. Same thing, gene therapy can correct on average. And this is for novel toy. This is for stranger marmoset. You can see they can all correct to the wild type level. Now, with this, we can, in mice, we tried very hard. Many other labs, Huda, many other labs tried to see whether you see a learning defect in marmoset. Actually, it was really hard to find. We actually, in our original publication, used water maze. Other thing we said we did not see, even in homozygous, we did not see a learning defect. That was published in our paper. But in marmoset, it's heterozygous. In humans, intellectual disability is a very, very severe problem. Actually, their average IQ is very hard to measure. I think 80 or 90% people you cannot measure, but the ones you can measure, the average IQ is about 30 or 40. So it's very low. So for marmoset, we can test whether we see a learning defect. So what we found is that you can use there are many cognitive testing you can do learning defect. So this is a very simple, right? Basically, 2-point choice. So basically, you touch the right place, you get a reward. You can see they learn very quickly, right? Y type. They make very few mistakes. But for Shank3, we found there are two phenomena. One is they insist to touch one spot. Like the marmoset will just go there and touch one spot, right? And so they just want to touch. So later on, we force them to touch more. Just if you keep touching the same place, you don't get a reward. So even with that, they still have preference on one corner, unlike a Y type. And they also make a lot of mistakes, right? So at the beginning, a lot more mistakes, right? So they learn much slower, and along the way, they more mistakes. So you can visualize this. Basically, this is touchpad. They have to touch this spot to get the reward. Y type do really well. Almost everything is in the middle, and they touch many, many, many times. Shank3, they know they need to touch, but they just touch the screen. They don't understand you really have to touch here. They also touch less times, so they do less trials. But with gene therapy, you can see you moved almost most of this into the middle, so that you can correct this kind of the defect. So to us, even more interesting to look at the circuit level changes, right? Because if you can permanently change the circuit, then you can predict that behavior will change, right? So one of the advantage of marmoset is their brain structure is quite similar to humans, so you can do functional MRI. This is anesthetized resting state functional MRI. So if you compare the local functional global functional connectivity, and you can see this is subtraction Y type from the Shank3. Red means Y type is higher. Blue means the mutant is higher. So in general, we have a feeling that the prefrontal cortex activity in the Shank3 marmoset is lower in Shank3, but the sensory part is higher actually in Shank3. Right? But if you do very strong statistical analysis, the statistical significance with a few marmoset is only this front part of reduced activity, reduced functional connectivity is very significant. And so you can use different views. And this was in collaboration with Fred in Bob Desmond's lab. And with the gene therapy, you can see, this is, I just showed you. If you compare Y type with gene therapy, Shank3 with gene therapy, you see this difference is gone. Right? So you can see that not only it can correct the behavior, but it can also correct the resting state functional connectivity in this animal, whether it's local functional connectivity or global functional connectivity. So you can see. So we are really get excited about this kind of stuff. So with this, both mouse and especially the monkey study, actually this thing is now in clinical trial. Actually, they just dosed the first patient in February. So we don't know the result. The hope is this will really help patient in the long run. This is the first try of the gene therapy for the Shank3, and we're hoping that with the clinical feedback, either is effective or have some effect, then we can continue to improve this kind of therapy. Because these patients are very severely disabled. None of them can really live independently. So in my view, many of these monogenic disorders, we can develop different gene therapy approaches in the future, and the most difficult part is how to get a new vector that you can IV injection and with good brain coverage, which now several labs and companies made progress. The other is these are rare genetic disorders. Most farmers wouldn't, invest. How to get, society to invest in this type of rare genetic disorder? Because these are one of the few cases we can really make progress. So we learned a lot from this marmoset Shank3 model, so we started thinking what else we can do to help the research community and the biotech community can generate other models that help us to test therapeutics in these models. So now we generated the MECP2 models, which is a human mutation arginine 276 to become a stop codon. Because there are many labs and many programs, this is also a terrible disorder, affect almost exclusively girls because I think boys cannot survive. And so there are many gene therapies, drug development for Rett syndrome. So we generated this model. Hopefully this will be useful in the future. We also generated present very early onset mutation, P1117S and P117L mutation. These patients become very severe at 30 years old. And 30 years old, about 4 years old, marmoset. So it's just, you don't have to wait for 20 years to study all this kind. So our hope is this will give us some insight. Alfonso survey made, now that was here, but now at Pittsburgh, also made different mutations, which is later onset, I think, and already show there are plaques in this marmoset, right? So usually knock-in a point mutation in mice, you don't see this kind of plaque and changes. So we are hopeful. With the Stanley Center, a consortium of the global consortium on the schizophrenia whole exome sequencing, identified this is the first publication in Nature by TJ and from Stanley Center, but it's a global consortium, identified 10 high penetrant rare genetic mutations contributed to schizophrenia, and one of them is D1A. So now we generate D1A because it was always difficult to generate models for study schizophrenia, and we initially generated a few mouse models to test it. In heterozygous, you can actually see some of the interesting phenotypes. So now we generated the first marmoset model. Hopefully this will also be useful. I think all the genome engineering technology, whether it's primate editing, genome editing, knockout, small knock-ins, are we made it very effective in marmoset thanks to the brain initiative and NIH support for genome engineering in non-human primate program. So, but as I said, most of the disorders in mental health, is, including schizophrenia, depression, autism, they are not monogenic. So can we use circuit dysfunction as a biomarker and as a testing for therapeutics, can identify things that could potentially translatable in the human? So we are focusing mostly on how do we identify circuits that behavior-triggered defect. I showed you in marmoset, and people already published in mouse Shank3 models. You can do resting state, which is anesthetized animal. But is this really same or different from the awake behavior triggered? Because most of behavior abnormality in autism is when they have social interaction or new environment or something irritated them, right? So can we test a behavior triggered circuit activity and whether they have a difference? So my lab always focused on corticostriatal thalamocortical circuits, and because of conservation, but also because it's implicated in many disorders. So in the last few years, we're still doing functional MRI with an assay animal, but we are more interested in now use this relatively new technology called functional ultrasound imaging, because it has a higher resolution, also temporal resolution and spatial resolution, but it also can do in awake animals. Sometimes you can do behaving animals because it's a probe on the head. So this is basically similar to, it's basically blood flow, Doppler imaging. And it's similar to functional MRI, you look at the blood flow to the representative of the correlated with the neuronal activity. So we tested this initially in anesthetized resting state, because resting state fMRI has been done multiple times, reported in Shank3 animal. And we know it's reported there is a reduced functional connectivity in the prefrontal cortex, in the striatum, and we show that actually in frontal cortex in the marmoset too. is reduced activity in SHANK3. So we tested, we do find a similar phenomena in the SHANK3 mice. These are about 10 animals for each. You can see Y type, and you can see that it's a relative activity, right? You can see a knockout is reduced from frontal cortex and to the striatum. So this is confirmed that this approach, this new technology, can be used to detect circuit activity changes, right, in mouse models. So then we designed an experiment to look at behavior triggered. So basically, what we did was to make the head fix of this animal, testing animal. Then we have a probe animal. You have a loop of tube. So the animal can come over social, then it could go away. So you have windows of social, non-social, social, non-social. So you can see when they are social, these boxes are social, you see the activity increase in the front cortex and in the striatum, right? So then you can do the same thing with a knockout animal. To our surprise, in the awake condition, every time there is social activity, their front cortex and striatal activity dramatically increase. So previously we were thinking based on electrophysiology in slice, based on functional MRI resting state, we need to boost the corticostriatal synapse. Now with this, what we need actually is to calm down the corticostriatal function. So this is not one imaging. This is the average imaging from about 10 animals. You can see the very significant increase, especially in orbital front cortex and in the striatum. So this become a new kind of angle to look, not only look at the circuits, but also to look at what kind of cell type is changed. But more important to us, can we find a target that actually can calm down these circuits of these behavior-triggered circuits neuronal hyperactivity? Then if we can correct that, maybe we'll eliminate or alleviate most of this behavior defect. So we found a target. We use NR2B modulator because the expression patterns and they are functioning in GABAergic neurons, actually. So what we found is that Y type, right? So you can see this before drug, knockout, before drug, you have hyperactivity. These are social triggered neurocircuit activity. We give one dose, just one injection. 2 hours later, activity, Y type have a little bit lower activity, but a knockout almost normalized to the Y type level. And one dose, 2 hours, 7 days later, it still has effect. It starts to creep back, but it still has. It's all very similar to Carl's ketamine. It's a long-lasting modulator of circuit activity. And so the prediction is if you can correct this so well at a circuit level, you should be able to predict that the behavior will change because these social behavior, these hyperactive are triggered by social interaction. So we did test them, use the same social assay, one dose, then you test 24 hours later and 21 days later. We also test 7. So you can see that it's very clearly, right, to change the behavior, that you can restore the social interaction. Very clear. This is black is Y type with vehicle, with drug, mutant with vehicle, with drug. You can see very clearly it can last very long time. Even 21 days later, it still has the effect. Similarly, for grooming behavior, one dose, right, top line, open circle is knockout with vehicle. The black open circle is Y type with vehicle. The filled circle is the knockout with the NR2B antagonist, and the black circle is the Y type with the drug. So you can see it lasts even almost 4 weeks. So we're really excited about this long-lasting effect, and we try to figure out what is the mechanism. Of course, drug is a drug. No compound is 100% specific. So we want to see, is this really NR2B? So we use CRISPR, we made a knockout, just adult knockout in the SHANK3 and animal directly. So this is control with the control guide RNA, scrambled guide RNA as a knockout. You can see with the social interaction, triggers the hyperactivity in the front cortex and striatum. But when you knock out the SHANK3 and NR2B, you don't see this hyperactivity. So we confirmed that NR2B is really the target. So now you all know block NMD receptor is probably not the best thing to do for kids or for adults. First of all, there are clinical studies show there are dissociative effect and many other problems, hyperactivity in animals at least. Right? In humans, it actually causes dissociation effect. So we are now very interested in, can we find the downstream signaling as a new drug target that could potentially really calm down this hyperactivity, behavior-triggered hyperactivity. We're also really interested how this thing could work so long. From drug is gone within about 24 hours. So why did the effect last so long? Is that affect LTP or LTD or something else? We actually really actively want to understand. We don't know yet. We have some hint, but we don't know yet. So our hope is that in the next couple of years, we will figure out the target, and this could be, you move on to testing in monkeys and in humans in the future. So I think I'm going to stop here. I want to acknowledge many people, especially our Marmoset team. This is not my lab's program for Marmoset. It's impossible to do. And we have many, many collaborators. It's a whole community effort, especially Bob Desmond, and Fen Zhang, Steve Hyman, Morgan Xian, and Ben Deverman at the Broad Institute. Fana Kreening, she was a postdoc with Steve McCarroll, now is an assistant professor at Princeton University, Steve McCarroll at Harvard, and then especially Broad and MIT Animal Facility. They take care of these animals so well, and every surgery, you need anesthesiologist or MRI scanner, scanning, anesthesiologist, surgeon assistant. Is better arranged than you go to hospital have surgery. I had several surgeries in the hospital, really well arranged, and they do phenomenal work to take care of these animals. And we have really collaborated Jim Pickle here, and also Alfonso Silva used to be here. And we also have our collaboration in Japan. Professor Okano and Sasaki, they are the pioneers of marmoset. They had a marmoset colony for almost 50 years. Asking me why 50 years ago you think a marmoset is good? I don't know. Actually, I asked the president of the CIEA. So they taught us a lot. We sent 3 or 4 batches of people there to learning the reproductive biology. So they are our long-term collaborators. And also we have collaborators in China. Some of my former graduate students, they're working on macaque monkeys. And we also learned a lot of reproductive biology of macaque monkeys to help us to establish the genetic engineering platform at MIT. So, and most importantly, I would really want to thank the many, many talented and dedicated animal cares. Each marmoset, unlike mice, you need a lot of care all the time. And we track their births on the cell phone. If they give birth, we have people, whether it's the middle of the night or in the morning, we will go there to make sure the babies are in good shape. So these are very, very dedicated people. And of course, without the financial support, none of them is possible. And we're really, I want to emphasize how the NIH Brain Initiative can help us, help many others in neuroscience field in the past decade. This is incredible technology development and model development. And we are really grateful to the support from NIH, but we're also very grateful for many of our donors, especially Hock Tan Lisa Yang Center for Autism Research, and also Lisa Yang Hock Tan Center for Molecular Therapeutics, Stanley Center from Stanley family, and Poitras family for the Poitras Center for Psychiatric Research, and many other NIMH and Common Fund, also NINDS, and also Broad Institute, and also the Brain Research Foundation actually was the first small foundation give us some money to get started for Shanksley Marmoset. Many also friends and donors for the McGovern Institute. Thank you.
Okay, thank you. That was, of course, an amazing talk, like we all expected. Are there questions? Yes.
Thanks for an incredible talk, just such inspiring work. Hi. Okay. 2 questions, if I could. The first one has to do with a genetic rescue in the marmoset. You have this beautiful multivariate characterization of their behavior. Did you find that some of the behavioral deficits associated with the deletion were more resistant to rescue than others?
Yeah.
And then the second question has to do with just a sort of broader question. how do you think about the prospect for inter-individual variation in genetics and environment inducing heterogeneity in kind of the variable penetrance of your manipulation when you put it into a real community?
Yeah, both are very good questions. The first question is there is a lot of variation. Not every animal is rescued at the same level, so we are as a group. But very obviously, very difficult cognitive testing, we cannot rescue. There's only a trend, right? And we don't know that's because developmental, you need earlier, or we don't know whether we have not enough neurons. Because high cognitive function is basically your brain computing power. We only got into 15 to 20% neurons. You may need much more, widespread. So we don't know. But there are things, mostly is the cognitive function. We cannot completely rescue. Simple cognitive function, we can rescue them. But the more difficult ones, we found that we cannot rescue completely. Yeah. And the second question is also a really good question. Although we give exactly genetic mutation, and some are, so it's a phenomenon that's really interesting. They all have defect, but they have different defect, and to a different degree. So if you use a computer, right, the cluster Y type, mutant. Then you bring new batches, say Y type, where they cluster? All the Y type cluster Y type. The new batch of mutant, they don't cluster to mutant. Because every mutant is different from Y type, but every mutant is also different from each other. So you can use the training to have a cluster, but the new one may not cluster into. So that means, so for example, some are very social deficit. Some are not that social they are okay, but the other things are there. So it's a spectrum of things, and kind of similar to human conditions. They are not, identical. Even they have identical mutations. Their just genetic background will affect them a lot, actually. So that's why I think that's why I mentioned comparing before and after is our most robust results.
Hi. So did I understand correctly when you said that by the functional ultrasound you find increased prefrontal activity in the Shank mutant, whereas all the old electrophysiology, slice physiology, and so forth told you that you should be expecting the opposite?
Yeah. So yes or no. So in the resting state and in slice, you find reduced synaptic functional connectivity. In slice, you find reduced corticostriatal.
Right.
But also, that's true. But in the functional ultrasound, if you do resting state, you also find reduced. It's only the behavior triggered activity is different.
I see. And you couldn't ask about the behavior triggered activity.
Yeah.
Because the question obviously is how this affects how we think about a huge amount of existing data that is sort of underlying our whole picture of the disease etiology. Right?
Yeah. So we also did 2-photon imaging in mice. If you use whisk triggering, they are also hyperactive. But if you anesthetize mice, they actually the picture may be different.
Okay. Thank you.
Okay, one question from online. This is from Suyin Lee. Could you tell us gene therapy at 3 months versus later? Do you see any differences in recovery?
Oh, that's also a very good question. So we 3 months seems like, since we started very young, it's a question a little difficult to answer because the marmoset behavior changes when they grow. So it's hard to say whether improvement is 9 months is better than 3 months because we cannot measure exactly same level. Like 3 months, their social level may be different from 9 months, even Y type. So it's really hard to say. So we don't have enough data to answer that question. But at 3 months after gene therapy, we already see very significant. So at the end, we put 3, 6, 9 months testing all together. So we don't see clear separation. We also don't see clear separation male and female. We also don't see clear separation because of the small number, whether 3 months injection or 9 months injection is better because you can all compare when it's 12 months. We did not see. For the functional MI study, actually it was way over 1 year, so it's a long-lasting effect.
Okay, one thing, I'm supposed to repeat the CME code. Sorry for the interruption. It's 57683. That's 57683. So we have one more question. And this question, I think a lot of us have this question. What is the threshold minimum number of neurons transduced by the AAV vector needed for effective rescue?
That's a very good question.
And also like the cell types.
Yeah. So that's a very good question and very tough to answer. Many others have done different studies. We did other studies. I think my feeling is if this is 15 to 20%, I think if you have 5% of neurons brain-wide, you will see a significant difference. And 10% definitely. We have local injections of 10%, we can see very significant change. And so it depends on the genes. For the SHANK3, if young, neurons are very plastic. They know they will shift their weight to the functional neurons instead of average. Right? So I think this is kind of, they will strengthen the synapses that can really work well. So I think 5 to 10% is probably will make a difference. Your other questions? I forgot.
Oh, okay. So have you looked at sleep disorders and their rescue in transgenic animals?
Neuron type.
Okay.
SHANK3 is expressing both excited neurons and inhibited neurons because both have excited synapse, and these excited synapses both need. So we use the synapsin promoter, so it goes to both types. And they match, the percentage match the endogenous. We tested this promoter. Sorry.
Oh, hi. Wonderful talk. Really shows the value of construct validity. And you had talked about some of the rare mutations that are associated with schizophrenia. I'm wondering if you guys are thinking about ways to model copy number variants.
Yeah, so very good question. So when Josh Gordon was here, he was very interested in the 22q deletion. You can model them. We can do large deletion, no problem. Duplication is more difficult. And so we tested that for, I think, 6 KB, 3 megabase, the smaller deletion. It works well in embryos, but we did not generate the monkey marmoset model because 22q also have a cardiac issue. And we were worried that the monkey will not survive, so we did not. So technologically, yes, we can do CMVs.
Okay, so one online question about sleep disorder phenotypes. Do you see any?
Yeah, that's, yeah. So we did a test, and we did not see, because we have this video, we did not see any significant defect in sleep. We don't know whether it's monkeys are different from human. We don't know whether it's because in the house conditions, they don't have naturalistic sleep. You have 12 hour on, 12 hour off, right? So that's, we don't know which is the cause, whether it's because of human-monkey difference or whether it's because the housing condition difference. We don't know. But we did not see, we did test it. We did not see sleep difference.
Last question from Yigu. She asked, are all SHANK3 mutations for autism loss of function?
So far, yes. Most of them are deletions, actually. Yeah.
Okay, that's it. Thank you, everyone, for your time.
Thank you.